Hello students, welcome to Read Med Prep Academy channel. Today we are going to discuss in Molecular Genetics Part 7 about the transfer RNA, the adapter molecule. Why transfer RNA is called the adapter molecule? The transfer RNA is a molecule of a cell that acts as a vehicle that picks up the amino acids scattered throughout the cytoplasm. It also reads the specific codes of the messenger RNA molecules. Hence, it is called the adapter molecule. This term was postulated by Francis Crick. Here you can see in this image the transfer RNA picks up the amino acid that is present in the cytoplasm and brings it to the ribosome and matches the anticodon with the codon on the messenger RNA, fixes the amino acids, forms the peptide bond between the amino acids, and it is again released into the cytoplasm to continue its work. The two-dimensional cloverleaf model of transfer RNA was proposed by Robert Holley. The secondary structure of transfer RNA looks like a clover leaf. In actual structure, the transfer RNA is a compact molecule which looks like an inverted L. Here in the Holley's two-dimensional clover leaf model of transfer RNA, you can see a D loop on the left side with the stem. It is called DHU, dihydroxyuridine loop. And you have the anticodon loop in the lower part. And on the right side, you can see the T loop, the T pi C loop, which consists of thymidine, pseudouridine, and cytosine. In between the anticodon loop and the T loop is the variable loop, and on the side of the T loop is the 3'N, which has a amino acid attachment site. What are the arms of the transfer RNA? The clover leaf model of transfer RNA shows the presence of three arms, namely DHU arm, the anticodon arm, and the T pi C arm. These arms have loops such as amino acyl binding loop, the anticodon loop, and the ribosomal binding loop at their ends. In addition, it also shows a small lump called the variable loop. Now let us go one by one. The D arm. The DHU or the D arm consists of a stem and loop with unusual pyrimidine nucleotide, 4 base pair stem with a loop contain dihydrouridine. Recognition site for the specific enzyme amino acyl transfer RNA synthetase that activate the amino acid. This plays a very important role in the stabilization of the transfer RNA's tertiary structure. Now the T pi C arm. The T pi C arm is named for the presence of the sequence T pi C. T for thymine, pi for pseudouridine, and C for cytosine, where pseudouridine is unusual base. This arm also consists of a stem and a loop. Stem contains 5 base pairs. Outermost of these pairs is CG. Loop contains 7 unpaired nucleotides. This loop contains a ribosome recognition site. Now anticodon loop. This arm also contains the stem and the loop. Stem consists of 5 base pairs, and loop called as anticodon loop or loop 2 contains 7 unpaired nucleotides. Out of these 7 unpaired nucleotides, the middle 3 form the anticodon. The anticodon recognizes the codon of the messenger RNA and binds to it. Here you can see the variable loop between the anticodon loop and the T pi C loop. Something about the variable arm. The variable arm has between 3 and 21 nucleotides, depending upon which the amino acid the transfer RNA encodes. Between the anticodon loop and the T pi U loop, this transfer RNA's variable arm is very short, so it looks quite different from other arms of the molecule. May present or absent, it depends upon the species. The length of the variable arm is important in the recognition of the amino acyl transfer RNA synthetase for the transfer RNA. Variable arm helps in stability of the transfer RNA. Transfer RNAs are called class 1 if they lack it and class 2 if they have it. Now the ends of the transfer RNA. the amino acid is attached to one end, amino acid acceptor N. The other end consists of 3 anticodon nucleotides. The anticodon pairs with the codon in the messenger RNA, ensuring that the correct amino acid is incorporated in the growing polypeptide chain. Here you can see the ends, the anticodon end, the 3 base sequence called the anticodon helps in recognizing the codon for its specific amino acid on the messenger RNA during protein synthesis. At the 3' end, it is the amino acid attachment site. An ester bond is formed between the transfer RNA and its specific amino acid to form amino acid transfer RNA during protein synthesis. The amino acid acceptor arm, the double helical, both 5' and 3' ends of the transfer RNA are present. 7 base pairs unpaired. At 3' end, 5' CCA 3' protrudes with the hydroxyl at the tip. It is a site for attachment of the amino acid. COOH of the specific amino acid joins with OH of the A in CCA to form amino acid transfer RNA. 4 different regions of the double-stranded RNA are formed during the folding process. Modified bases are especially common in transfer RNA. Wobbling in transfer RNA. Wobbling between anticodon and codon allows some transfer RNA molecules to read more than one codon. Here you can see the wobble. The transfer RNA anticodon UUU can bind either to the messenger RNA codons of AAA or AAG. The third codon is called the wobble codon or a wobble position. Both these codons code for the amino acid lysine. Now something about the charging of the transfer RNA. The process of addition of amino acid to the transfer RNA is known as amino acylation or charging, and the resultant product is called amino acid transfer RNA or charged transfer RNA. Without amino acylation, transfer RNA is known as uncharged transfer RNA. Here you can see amino acid transfer RNA synthetase, which is an enzyme, combines with the amino acid in the presence of ATP and adenine. And this forms a complex amino acid adenylate acid. And this combines with the transfer RNA and releases the phosphate bonds, releasing the energy for the charging of the transfer RNA. And the amino acid transfer RNA synthesis is released along with the adenine and the phosphates. Amino acid transfer RNA synthetase reaction. Amino acid charged by addition of AMP from ATP. Subsequently, hydrolysis of PPI makes this irreversible. Amino acid transferred to transfer RNA and AMP is released. You can see the reaction on the right side where the amino acid combines with the ATP and there is a release of energy, and the combination of the amino acid along with the ribose and the adenine, and amino acid adenylate is formed, and this amino acid adenylate along with the transfer RNA, there is release of AMP and the amino acid transfer RNA is formed, and the transfer RNA becomes charged carrying the amino acid. If 2 such transfer RNAs are brought together, peptide bond formation is favored energetically. Number of amino acids are joined by a peptide bonds to form a polypeptide chain. Here you can see that the transfer RNA carries the free amino acid that is present in the cytoplasm according to the codon and brings it and matches it with the codon that is present on the messenger RNA in the ribosome, and as the charged transfer RNA attaches itself to the messenger RNA, the amino acid peptide bond is formed along with the previously formed amino acids, and once the peptide bond is formed, the transfer RNA is released and becomes uncharged into the cytoplasm. This amino acylation is catalyzed by an enzyme, amino acid transfer RNA synthetase. This is an endothermic reaction and is associated with the ATP hydrolysis. Here you can see that the amino acid transfer RNA synthetase combines with the amino acid along with ATP and releases the phosphates, and becomes amino acyl adenylate, combines with the transfer RNA and releases the energy AMP and utilizes the energy for the charging of the transfer RNA, and the amino acyl transfer RNA synthetase is released and the transfer RNA is charged, and this transfer RNA along with the elongation factors comes to the messenger RNA and elongates the peptide chain. 20 different amino acid transfer RNA synthetases are known. The power to recognize the codon on the messenger RNA lies in the transfer RNA and not in the attached amino acid molecule. So today in molecular genetics part 7, we have discussed about the transfer RNA, which is the adapter molecule. Thank you. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.